Okay, yeah, I was going to have to pretend to be Maurice, but you saved me. Okay, so welcome back, folks. We have our final talk before we get into our panel discussion from Maurice Whelan, who's with the European Commission, the Joint Research Center, and he's going to talk about experiences with paradigm change in European Union science and regulatory decisions.
Thank you very much. It's a great pleasure to be here. My thanks to the organisers for inviting me. I've really found yesterday and this morning's presentations, discussions very, very stimulating. Yes, I'm going to have this problem too. I'm pressing the green arrow button. I'm pressing, is it? Yeah. Okay. So yes, I'm going to try to give you a little flavour of what has been going on in Europe for the last 10 years or so in 20 minutes. So bear with me. I've cherry-picked a few things. This is going to... So let's talk about the drivers. This is a very significant driver that we have in Europe. It is the Directive for the Protection of Animals Used for Scientific Purposes. And scientific purposes, we mean it also includes testing for toxicological hazard assessment. But also this directive deals with the use of animals for basic and applied and translational research. But look, the bottom line is there's a policy goal that eventually within the EU we want to move completely away from animal testing. Key phrase here at the bottom is as soon as it's scientifically possible to do so. You know, but that's the trajectory we're on. So that's very much an overarching policy in the EU. The duties and mandate of the European Union Reference Laboratory for Alternatives to Animal Testing, better known as ECVAM, that I'm responsible for also, is, you know, the duties are outlined in the directive. And really we cover everything from, you know, participating in research and development activities through validation, dissemination and promoting international dialogue for acceptance and uptake. Just well, their validation is a very important part of what we do. In fact, the European Centre for the Validation of Alternative Methods was the original, let's say, activity housed in the JRC, which is now over 25 years old, or 25 years of activity. You know, I truly believe that validation is essential, a part of the process for gaining acceptance and uptake, but it's not sufficient. And also I think we have to be as innovative in the way we validate as we do in developing the methods in the first place. So I'm very happy to be working very closely with NICETM and Anna and many others to really see how we can, as I say, be smart in the way we approach validation. I often talk about the 3 Ps of validation: principles, you know, purpose, and practice or process. And I think we have to be clever in how we design validation strategies to make sure we get the best out of them. This is our status report that we publish every year. Actually, this is going to be coming out next week, I hope, when I get back to the office. I hope it's all there ready. And I'd encourage you to go and read that if you're interested in develop validation and acceptance of methods. Of course, the other main driver for us wanting to transition to this new paradigm is all the legislation that we have. You know, there are over 100 pieces of legislation in the EU related to chemicals and risk management associated. So it's a bit of a nightmare in a sense to navigate. But there are some key ones here listed. First of all, the REACH and classification, label and packaging legislation, which are kind of horizontal pieces of legislation. And then you have the sectorial pieces of legislation like plant protection products or pesticides, biocidal products, antimicrobials, I think you call them over here, and cosmetics. And, you know, there are different you know, emphasis in these legislation about using alternative approaches. I mean, everything from the cosmetics sector, for example, we've had a ban on testing cosmetic ingredients since 2013, a marketing ban. In REACH, we have strong provision for use of alternatives. We have what we call Annex 11, which allows for what we call the adaptation of standard information requirements, encouraging the use of alternatives. Of course, there are more conservative or more restrictive pieces of legislation, like the pesticides that Anna referred to where, the provisions are more explicit. But also we have, emerging challenges. We have endocrine story also in the EU. As you know, there's been a long debate, continuing debate about actually defining criteria, or scientific-based criteria, for what constitutes an endocrine disruptor. But, generally speaking, we anticipate that those criteria will be linked back to the WHO definition, that it would have to have an observed adverse effect in the whole organism, should demonstrated that the mode of action is endocrine related, and both those things are associated with each other. In the mixtures area too, the risk assessment of combined exposures and so on is very challenging. So here, of course, the emphasis on reduction of animal testing is important, but very important too is the fact that honestly, I don't think we can address these policy goals if we don't embrace new approaches and new technologies. Generally speaking, our risk assessment paradigm it relies on generic risk considerations, let's say what you would say more hazard-based considerations, but also specific risk assessment. I think it's important, though, that here we've been emphasizing a lot the protection of human health in the environment, which is, of course, paramount, and we truly believe that shifting to new approaches will enhance that, we hope. But let's not forget too the other two aspects of chemical legislation, particularly in the EU, is we need legislation that promotes innovation, competitiveness of our industries, and of course sustainability. A circular economy, we call it in the EU. And also, we need to ensure that, markets function. The EU is primarily, it's a single market concept that really gave birth to the EU. And of course, international trade, fundamentally important. So we need to keep those three things in mind when we talk about what the success looked like in transitioning to a new paradigm. Some of you might agree with me, some of you mightn't, but back in 2007, obviously we had this pivotal report, seminal report, and made a huge impact worldwide, particularly also in the EU. In fact, I changed the direction of my career after reading that report. God help me, but anyway, too late now, 10 years on. But yeah, for better or for worse. But I personally believe that in that 10 years, what was essentially a technology push or a science push has transitioned into a pull from the regulatory world. And that's very, very important. I do think we're past the tipping point in that sense. And, there are a number of organizations leading the way. Obviously, the US EPA here in the US. In 2013, the scientific committees advising the Commission basically, regurgitated a lot what was said in the 2007 NRC report, but basically made it formal in the sense that the Commission should follow this new paradigm. And the agencies, particularly the Food Safety Authority, ECHA, the Medicines Agency, all have very active programs internally for exploring the use of new approaches. So it's not a question of if, but really how and when. And then, of course, the OECD, fundamentally important. I have the impression sometimes a lot of people think the OECD is some kind of independent, distant organization. The OECD is an organization of members. We're all members of the OECD. And, I think that's important because, I know it's a cliche, but it's true, that we can make progress through collaboration. And using platforms like the OECD for working closer together, and the good news is, look, we're making a hell of a lot of progress. When you look back over the last 5 years, 3 years, important things. That over half of the program or projects at OECD now in the test guidelines program are related to new approaches, to non-animal approaches, over half. We've got tools like the QSAR toolbox emerging. I've had the pleasure of co-chairing the AOP development program along with Bob Cavlock up to recently and his recent retirement. We have emerging frameworks, integrated approaches to testing, assessment, and defined approaches. I know it's more acronyms, and I know it's all kind of terminology, but it's important because we all know that, you know, collaboration and working together depends a lot on having harmonized vocabulary, harmonized terminology, and this is important. We've got these case study projects going on now that really, you know, rubber hits the road. You know, you get from theory into practice, and agencies can share their experience with real, you know, chemical risk assessment scenarios and show how it worked, what didn't work, and so on. Very powerful. And of course, a whole slew of different guidance on how to use new approaches and so on. So I strongly encourage, you know, I have noticed myself though when I'm there and working, you know, if countries don't send the right people, if they're not committed to those programs, then you know, there'll be a limit of what you get out of them. So it's important that member countries do commit to these programs and are aware. Let me jump back to the REACH, because REACH has, and the chemicals legislation in general depends a lot on products of from OECD, and you know, it's helped a lot in the use of alternatives under registration in REACH. What you're seeing here, you don't need to get into all the details, but what you're seeing here is a summary of you know, the data sources used for registrations from 2008 to 2016. It's about, you know, just a bit over 6,000 unique substances. And basically what you see is that, you know, new experimental studies for the purpose of satisfying information requirements were only about 10 or 11%. So very little new studies. A lot of existing data used, which is good. But then you see read across in particular, and a bit of QSAR being very strong. So you could say that the most popular alternative approach for chemical assessment in Europe at the moment under the REACH program is grouping read across. As I say, and you see a lot of waving as well. Now, you can look at this both ways. You know, one, it's good news. We're using a lot of alternative approaches. On the other hand, we have to be careful because this information is just what's registered. It doesn't necessarily say that those registrations or that information set is sufficient. So in fact, when you look at a lot of the analysis of the read across, there's a lot of non-compliance. In other words, there's a lot of nonsense in there, you know. So, you know, again, this is, you know, we have to be responsible in terms of how we transition new approaches. We want to ensure that level of protection and avoid animal testing. Anna touched on the skin sensitization. I just wanted to mention it briefly too, because it does represent a very significant, I think, success for a number of reasons. So this is the actual legal text from the REACH Annex 7 that specifies that in vitro methods are now the new default route to provide information for skin sensitization under REACH. So you could argue full replacement in a sense, as long as your chemical falls into the applicability domain. But what are other important things about this? First of all, it represents, you know, if you like, a legal acknowledgement that this is not a one-to-one. So we're talking about the replacement of one test guideline or one kind of test with multiple in vitro tests. So that's now, you know, as scientists, I know that's a no-brainer, but when you see it written into legislation or reflected in legislation, that's important. That's paradigm shifting happening. Another key point here is the fact that the in vitro methods or the information requirements are expressed in terms of key events. You can see them listed here: molecular interactions of skin proteins, inflammatory response, activation. You know, that's fundamental to our paradigm shift, is we want to move away from expressing, you know, hazard in terms of the ICTs, the apical endpoints, and actually talk much more mechanistically. So here it is actually spelled out. I mean, there's a lot behind this, and we published a recommendation in early this year, just to spell it out for stakeholders, for industry, and so on, about how to go about this. So I encourage you to have a look at that. But you know, this is a culmination of many, many years of work by many people, lots based here in the US, and a lot of hard work at the OECD. Interestingly enough, however, even with this, there was a lot of pushback from industry and from regulators saying, yeah, but how do you combine the three methods? What's the prediction model? Could you write that into the legislation as well? So although I appreciate Stan's comments from yesterday about you know, flexibility and fitness for purpose, and you know, as a scientist, I'm all for that. But when you get into this level, people love prescriptiveness because what they want certainty. So in other words, they want to know that if I submit something to an agency, there's a very good chance, and better if I get sued, you know, there's a better chance that this will stand up. So, we are now working at OECD to see, can we actually capture this defined approach in a guideline, a new instrument that would have the same regulatory footing, the same footing as a traditional test guideline. Another key point in bringing that skin sensitization thing over the line was finally having a discussion about the elephant in the room with the regulatory committees was, listen, here is the variability of the LNA data. Let's all be, honest with each other that, it has offered, good levels of protection, although there's an increase in skin sensitization issues, particularly in occupational context. But anyway, reasonable levels of protection. But from the clinical data that we have, from the human data we have, we know it's not perfect, that's for sure. And there's a hell of a lot of variability. So we were able to take that into account when we were then proposing this alternative approach. So the idea wasn't proposing a perfect solution. The idea was proposing a solution that was at least the same, if not better, and we would argue that it's better than the traditional approach, which I think highlights the importance of something that we've really neglected for many, many years, is appreciating that, in fact, understanding uncertainty, describing it, talking about it, is extremely important for, moving towards people using new approaches. Because a lot of the question is, it's not about how perfect your method is. It's like how wrong would I get my decision, if I start using this and this is evidence, because these are sources of uncertainty associated with animal tests, and there's a lot of them, isn't there? But I call them sources of familiar uncertainty, because people are familiar with them. They can begin to work with them. We can add on safety factors, and we can, make provisions in our decisions and so on. So I think what's incredibly important as we go forward is to start talking about the uncertainty associated with not just the inputs to our risk assessment. What is the uncertainty of using a human-derived, cell in an assay? And what's the uncertainty in that representing human biology? I know it might be human-derived, but does it represent the pathways that we're interested in and so on? What about the uncertainties with computational chemistry? Can we describe those in a way that decision makers... And of course, we're also getting more and more sophisticated ways of combining all this information. What's the uncertainties associated with that? The good news is we don't have to start from scratch. There's an awful lot of very, very rigorous scientific-based work being done on how to go about describing uncertainties and present it in a way where end users and so on can begin to grasp it. So I do think we can do this, but we definitely need to commit to doing it. I just want to sort of end of my talk is talk about a thing called scientific credibility, which we're all very interested in. And, as I said, we have 25 years experience in doing validations and so on, but I started to think and getting very philosophical in my old age about, how it's more than just validation, isn't it? It's about scientific credibility. And, this is a definition of credibility which comes from, 1980, and actually nothing to do with toxicology. There are a lot of fields where prediction models are used to inform decision-making, and decisions that have serious socioeconomic consequences. And the question is, how was confidence built in those predictions and those models? In the nuclear industry, aircraft industry, all these other sectors that maybe we're not too familiar with. But people have been using predictions and models for a long, long time. Important about this whole scientific credibility is actually the process. That essentially it's a social epistemology process, meaning, it's about developing or striving for a shared knowledge and understanding between all key actors. So keep that in mind as we go on. Now, another good news is, there's a lot of smart people have been thinking about this. They've been thinking about the value of scientific theory, thinking about the criteria that one uses to pick a theory for your decision-making process. Thomas Kuhn or Macmillan, these way back, 70s and 80s. They're philosophers, and there are a lot of philosophers here during this meeting, on the philosophy of science. And so you can draw on a lot of that very learned work. And this is our kind of our attempt to try to begin to bring this into a predictive toxicology aspect. And what this is a credibility matrix. And imagine it as a way of having a good discussion, okay, about credibility. And you can basically define the credibility in two dimensions, two axes. One is the strength of the knowledge underpinning your predictive approach, and the other is the strength of the data. And I define strength here as meaning, availability, applicability, and dependability of either the knowledge or the data, okay? And what you can begin to do is you can say, okay, can I orientate myself with my predictive approach in this matrix? So, for example, if I have a predictive approach, and I mean predictive, it can be a combination of in vitro and in silico and whatever, very generally speaking. If that's based on really very sound biological-based reasoning, I really know this toxicological pathway or whatever. And also have lots of data to compare with, my predictions with what I'm actually trying to predict, okay? I would be down in this dark left green, highly credible zone, And ideally, that's where we'd like to be, of course, super confident. If you're up in the white corner opposite, that's kind of the Wild West. Therefore, you've got to be careful about using that model in your decision making because you've no idea of the principles underpinning it and you've no data to confirm. But then you can navigate around because you can see that, in a sense, people's credibility, judgment of credibility will be a mixture of those two things. So, for example, imagine if, I really don't fully understand all the biology, don't fully understand mechanisms, but I have a lot of data, in terms of being able to see how predictive my approaches are. And, that's, for example, a lot of that kind of modeling is done in stock markets and so on, where you've got loads and loads of data, but you don't fully understand what's going on. The other extreme, and the one I suppose that, let's say, is becoming more and more relevant for us, is where you will never be in a position to be able to compare your prediction with what you're actually concerned about. For example, the effect of chemicals on human health. How good are we actually at being able to measure that? So in a situation where we mightn't ever actually be able to validate quantitatively, we'll have to begin to rely much more on our belief and our understanding that a predictive approach is relevant. Take the area of developmental neurotoxicity, for example. We're trying to bring in vitro methods into play in pesticide assessments. And there, we don't have enough data on developmental neurotoxins to do a kind of statistical predictivity thing, The way we'll get acceptance is really to say, listen, we understand, the biological processes underpinning the development of the human brain. And here are these key processes, neurite outgrowth, etc. And we can capture those in vitro assays. So if a chemical is really hitting that process hard, I would be concerned about that for biological reasons, okay? So I'd be shifting the discussion now to what I call an epistemic validation, thinking about the knowledge underpinning those processes as opposed to some data you have in predictive performance. You can begin to codify these into credibility factors. I won't go into it. But again, I just want to emphasize, by doing this now systematically, giving us a scientific-based framework on where, with which we can reason about credibility and our expectations, you can begin to, see that it's not just about the predictive performance of a method that actually leads to building confidence and credibility. And as I say, the process is important as the result. And again, this was touched upon, I think, in Stan's talk, in Anna's talk, and others, about this whole idea. I described this credibility matrix. This isn't about absolutely being able to put things in boxes. This is about, that twister game where you all fall around? Think of it like that. So it's not about absolute. It's about agreeing on where you think we are, and then deciding if that's good enough, or do you need to go to another box? And how will we go on that from one box to another? Will we take a knowledge route? Do we need to understand more? Or do we take a data route? Maybe we could generate some data and help us. So you need to understand it in that context. And I think, when we talk about the evolution of validation and process and so on, this is kind of where we're at now, is how can we embrace a lot of what was said in the earlier talks and really bring it into play. And case studies, for example, are a good vehicle in this context, So now I'm going to be really brave and say, okay, look, if we're talking about paradigm shifting, where are we going? How far are we willing to shift? And, back to the 2007 report, I asked myself a lot, about what should we really be trying to predict? Should we really be trying to predict the result of a 90-day study? Because really we need to be asking ourselves what predictions actually give us the level of protection we want to achieve. And then you can't help ask yourself, well, where are we going with this? Because maybe we're actually talking about, redefining toxicological hazard in a more appropriate way. Talking about the key events that I mentioned in the skin sensitization context, where we begin to articulate information requirements for toxicological hazard that we will use in a risk assessment in terms that are more relevant, that are more suited to actually the tools we're using to generate the information. If I keep articulating an information requirement in terms of the results of a 90-day rodent study, that's a square peg in a round. That doesn't fit with all of the type of new tech that's coming along. So we need to bridge that gap. And what kind of brings me then to conclusion is, what are we talking about? Are we talking about replacing animal testing, or maybe a better term is displacing animal testing? That basically it's about, let's take our eye away from the animal test for the moment and think about we're bringing in a whole new way of doing business, a whole new toolbox, a whole new way of thinking about the problem, and ultimately a whole new way of making decisions. And in that sense, we're seeing a sort of a move to a whole other paradigm at all levels. We're generating so much data now on alternative approaches. Even the most conservative guy can't ignore this, because if we have mountains of data, people will be analyzing it and they will want to be doing something with it, I talked about the information. If we know much more about biology and toxicology, it's inevitable that knowledge will inform, a revision, an evolution of the information that we seek out to make good decisions about. And as I say, the ultimate game, of course, is to make better decisions, wise decisions. But in this new paradigm, another incredibly important thing is that we learn along the way. We're always learning. And you can't say that's, let's just say, a strong feature in our current paradigms today. We don't learn very much from the regulatory animal tests that we do. And of course, if we're really talking about paradigm shift, let's face it, in the next 10 years, we'll be displaced by all these young people. We ran a summer school in May early this year, and over 100 people turned up. I couldn't believe it. But my God, the energy and the enthusiasm. And then you realize that, this is as important a vehicle as any kind of case study or whatever, because, as I say, in 10 years' time or so, these are the decision makers of the future. And if we get all those things right, I do truly believe, that we're on the path to this big win, that we can replace animal tests. But we'll do that in a way where we can ensure the highest level of protection for human health and the environment. And we can do that and build a strong economy too. So thank you very much.